where you can see. Oh, that's the other way around. So this is actually the other way around. So that's a bad slide. The slide that we're looking for, the slide what I was looking for, FST is high. So I thought I was going to make an example that sometimes you look at places that FST is low, and then you can find things like that.
What does that mean?
That means that the individual differences are higher. So HLA would be a place was a signature like that. So there's a selection for diversity. And well, actually, one of the reasons for this is that way to avoid these kind of problems in different signatures is combining them and put all the, look at all kinds of signatures at the same time. Here's the one that's been commonly done with population genetics now when they plot FST against heterozygosity. And there is a genome-wide expectation that FST versus heterozygosity is going to be a certain level. And then you start looking at outliers where their heterozygosity is low and FST is high, and that's what we want to see. Heterozygosity low, FST high. Small variation, big difference. And this is what we did last year with another of Steve's former postdocs, Aug. So we published this paper when we thought, wouldn't that be cool to go to Africa? Well, we didn't go to Africa. We went to Africa after we published it. And think about diseases in Africa. And we know that diseases like viruses like HIV came from Africa. And the populations in Africa probably were exposed to this virus for a long time. About HIV, it's been a lot of literature lately talking about how it came probably from people that were hunting chimpanzees or other primates, and we thought, well, that and it probably came from them while they were working with them, they cut their hand or whatever other way. Hopefully not by having sex with them. And this practice of hunting primates was probably happened for a long time because people lived in these regions for a long time. They probably hunted them a long time, and they probably got infected many times. And if they got infected many times, that would be an interesting speculation to say that these peoples may have been by now adapted to these infections. So maybe that they've been exposed to this selection pressure for a long time, a lot longer than everybody else. What if we can look at these populations that still hunt chimpanzees and compare them to their neighbors and see if we can find a selection signature in the genes that we know today help you survive HIV infection. So we looked at the two populations here of pygmies, one called Mbuti and the other one's called Biaka. And the Biaka ones are known to still hunt chimpanzees. Right? And then they also look at the other African populations of which we had available genome sequences from HapMap and 1000 Genome Project. And these would be the Bantus that were here, the Mandinkas, and the Yorubas. And we compare them in the 5-way comparison. And so we compared every gene in the genome to create an average expectation. And then we looked at where in that expectation will be the AIDS restriction genes, would be the genes that are involved in protection against HIV infection. And we came up with 2 of them that were on the very top of this distribution. The very, very top of this. They had very, very high high reduction in heterozygosity. That was completely out of the woods. You see? And these are some of the genes that are today involved in African population in protection against AIDS infection. So at least that works, and we published that in DMC Evolution of Biology that came out end of last year. Made a lot of press as well. It was paid for by Bill Gates' exploration grant. So this is good because this was the first paper that used our method and showed how nicely it can work. Lots of people, however, today like this other approach, which is based on that tendency. Remember, I was telling you the tendency of recombination. So when you have chromosomes that are not selected recombine into the chromosomes that are selected. So remember, the first thing that's during selection, the whole chromosome gets selected. You select the whole chromosome, and then the region gets smaller and smaller and smaller. But if selection is strong, then it grabs a lot of chromosomes at once. So that region that we can detect to be selected is large. It tends to be large. And large in common, so we pick that. This is actually, can you see this? This is the selected chromosome, and that's a selected locus. Right? This is the first stage of reduction of heterozygosity, but it happens in the whole chromosome. Right? You grab the whole thing. And then the ends of these start being recombined into. However, you still see this sort of a blob, this sort of a group, these extended chromosomes from here. There are daughters, right? All of these chromosomes are daughters of this original chromosome, and they extend far away from the selected locus. So this, if we can identify this is another signature of selection. It's called extended linkage disequilibrium. Okay, and that's the pattern that you observe. You observe many long extended regions that are descended from the original chromosome. Now there's a mathematical step before that identifying these haplotypes. Haplotypes meaning sequences of alleles along the chromosome. So that is the part that's difficult to do this test, but you can you have to phase the chromosome first and then do this test. So sometimes it's hard to do, but if you do that, this signature of selection is very good at identifying a very recent occurring selection, because if it's recent, then your region is going to be large. Okay, so this is particularly my favorite gene. It's a gene for lactose tolerance. Okay, so how many people here can digest milk? Everybody? You can. You can. Can you? Can you digest milk?
Yeah.
You still can.
Are you choosing?
No, you can. So I can't.
You can.
No, I was talking sour. I think sour cream out of my borscht today. So please don't give it to me.
So you don't like to fund you?
Yeah. Well, there's a Nature issue 3 weeks ago that was dedicated just to that. And Northern Europe is 100% lactose tolerant, and Southern Europe is 50-50. And that's because of adaptation to living with cows. So people lived with cows, they made everything out of milk, and in order, remember this is differential reproduction, so you contributed a lot more because you're healthy and you can live longer and make more children because you can always drink milk. And when I was 30, stopped. I could not. Cheese, milk, butter, can't. Keeps me in good shape, but
So you drink soy milk, yeah?
I don't drink any milk. Any milk. If I can help it, I will do it. But it also means no chocolate.
But you can eat dark chocolate without eating.
Wally, are you lactose tolerant? You are. That's very rare. Okay? People from the east.
That means you had some kind of a European ancestor.
People from the east. If you, I could go to Chinese, Japanese restaurants, Thai restaurants, no problem, because everybody there cooks without milk. So I know I'm safe there. As soon as I go, and in America, American food is cooked based on believing that everybody's lactose tolerant. Because English, German, Swedes, French, they're all lactose tolerant.
How about yogurt?
Yogurt I can, because it's already broken down. I can yogurt, kefir. I tried kefir yesterday. Yes, I can do it. Yes.
But so in Swedes it's 100% recycling.
Swedish, Danes, Swedes, so northern parts. Because we could survive still in the south by growing agriculture, but in the north it was more for milk and cheese.
But it's not 100%. I knew one girl, she was my colleague, Welsh, she was lactose.
No, it's 95%, up to 95%.
So, a question here occurs. So is there a way to estimate the divergence time based on the size of these?
Yes, and people do that. Yeah, so the more recent it is, the bigger it is.
Yeah,
Right.
the correlation occurs, but
And what
they
about,
estimate it.
and what about heterozygous people with this?
No, this,
Is they okay?
I
So
think
they,
it's.
that was-
I think they have intermediate because you.
so they
So the problem,
what do they
it's a loss of function allele. So lactose gene is. This is not the one that's mutating. There are two different adaptations in Africa.
How long is the haplotype for lactose intolerance on average?
So this is 500 kb is this, so that would be 1500 kb.
1500 kb, okay. So that correlates back to about, I would say, 1200 years ago.
Which is rise of agriculture.
Alex and Andre, if you read the paper where we proposed Delta 32 was selected in 1996, there's a formula in there which relates the length of the haplotype to the time elapsed since the last selective pressure.
Yeah, thanks. That's what I wanted.
That's exactly the form. It was developed by two people. One was Clay Stevens, and the other one was David Reich. And David Goldstein was in that mix too. And Stevens and Reich fought for a year about the mathematical thing. I'd like you to take a look at this, Sergey, this equation. It's an algebraic equation that relates the length of the haplotype, the average recombination rate across megabases, and the time elapsed since. And that's where we came up, and it's using that equation that we came up with the 700 years for the age of Delta 32. That haplotype is about 1 megabase long. 3 different studies showed it. This one is 1.5 megabases. Delta 35 was about 700 years ago. That's why I said this thing is about 1,200 years ago, based upon that equation. If there are other equations out there, I haven't kept up with the literature, but I think, roughly speaking, it's pretty accurate.
Well, there was
You
another.
want to talk about other equations
You want to talk
that were
about
developed? Who developed them? Or is Reich's?
It's-
Reich and
it's
Stevens,
all
by the way,
about-
they fought for a year.
Yeah.
Finally, Clay backed down and said Reich was right. David Reich was 22 years old when he wrote this.
Yeah. You supported him then, so.
I did.
No, and it's still nice to me because he somehow connected to you. So there's another paper that had to do with published by another one of Steve's students. Looking at this, instead of people, they looked in wolves. I don't know if you remember this paper. This was there's a bunch of people here that you connected to. And Bob and Carlos and Bridget, they all from the same similar shop. And this is a really nice story where they look at you think about a wolf in Russia. What color is it?
Gray.
Gray, right? So that's obvious, right? Except for in America, it's not. But if you start looking at pictures of wolves running around in Alaska, you'll see those black ones, and you're like, what? That's not right? So, and Americans actually kind of like let it slide until they were moving wolves into populating Yellowstones, and they got a bunch of samples from there. And I think that's how the whole thing started. They started looking at this black allele and trying to figure out where is this coming from? Why do we have black wolves? They did, so this is KB allele that makes it black. So this is a young black wolf. It actually gets gray at the end. And this is KY allele that makes it kind of white at the end, which is the sort of normal looking wolf. And so what you will see around the KY versus KB being black, KY will be white. If you look at the heterozygosity, you'll see that the heterozygosity is kind of bouncing up and down for the Y, but for the B it goes down. So that's a selected allele. Right? That's one of the signatures of selection. But they were not really satisfied with this. They also looked at the extended haplotypes. And if you look at the KY allele, so this is not a way of looking at these haplotypes. If they're short, another way of thinking about it, that means they will break. So their sequence is not the same, which means you cannot identify the original chromosome. It's not the same chromosome. All these, this looks like a squashed bug, like an insect that you squashed. If you see this, it means that there is no pattern. Everybody's different, right? That's what we see. Everybody's different. So you start with the allele, and the next thing it could be the A or B, A or C, and the next thing could be G or T. So there's no one haplotype. KB allele has an extended haplotype because if you start with a KB, the next in every chromosome will be an A, and the next allele in every chromosome is going to be a T, and the next, which means extended. Everybody here is the progeny of that original chromosome. It keeps being selected. So this B allele is being selected. It's being selected in certain places. And if you look here, what you actually realize that these are in the forest and these are in the open. So that could be one of the things that in the forest it's dark, and if you're black, it's a good protective color, and you blend in. And then you look at the age of this allele, and it turns out to be very old. It seems to be
How old?
But it's
How old?
well, it's the allele that it exists before.
Wolves diverged
Before
from something?
wolves and dogs have diverged from each other.
Well, that's not that old. That's only 18,000, 15,000 years.
Right. Except when you look at that allele inside of this wolf population, it seems to be only Okay, I'm losing the line here. The point is that the allele is very old. When yeah, you're right. When wolves diverge from whatever the
Oh,
ancestor,
you mean from another canis species?
from an allocated species, but the
Coyotes.
allele in the wolves in America seems to be only 18,000 years old, which is the time when the dogs came to America. So these guys look at there and they show that this allele actually was brought by the dog, which admixed into the wolf, and now this allele is being selected, but it's not even from that
So back
animal.
from dog to wolf to wolf.
It jumped from the dog to the wolf and then started being selected in wolf because it had a selective element. So that was really cool.
And what did these austere authors propose was the selective pressure
On them in the
on the black allele?
Yeah, on the black allele.
What's the answer? What caused the black allele to have an advantage? Camouflage?
I thought it was camouflage.
Okay. You thought it was camouflage.
I thought-
Klaus?
That was one, but they're now, I mean, Bob has a graduate student working on, investigating this much more, because this K locus is also highly expressed in the immune system.
Right.
And so there's something, these wolves are responding to a different immunological pressure between tundra versus forest, actually. And that gene is also important for, it also seems to be expressed in a very particular part of the brain that actually controls pack behavior. So there's a lot going on here, and there's another graduate student who's focusing on this intensely to figure out functional genomics.
I heard that from Bob
Yeah.
this summer. Yes.
Yeah.
I recall that. Anyway, but their interpretation at the time was maybe it's camouflage.
Camouflage.
And I get the IUCN Species Bulletin newsletter, and the last issue there was a sort of
Survival thing. Yes.
yeah, and there was all these pictures of animal products that had been illegally traded, and the last issue, because Bridget saw it and I sent it to Bob as well, but there was a picture of hundreds of wolf skins from Russia that were confiscated at the Chinese border because they were coming from Russia to China. And among those skins were lots of black wolves. And they were wolves, they weren't dogs, they were black
So
wolves.
this also.
And so it might be, again, the same thing might have happened here in Russia where dogs admixed with wolves and introduced this black K locus. So we're trying to find out how to get some of these samples, to UCLA or to Bridget.
You're in Russia now.
Yeah. Well, I talked to somebody at the Russian Academy of Sciences. There's 2 wolf hunts every year. They kill thousands of wolves because, it's a lot.